So I'm Alyssa Novak, and I am research faculty at Boston University. And today what I'm going to do is tell you a little bit about seagrass biology, ecology, and share some of my experiences monitoring and working on restoration projects. So for those of you that aren't familiar with seagrasses, they are marine angiosperms, which means that they are flowering plants. And they grow submerged in marine and estuarine environments, and they grow from the intertidal zone to approximately 60 meters in depth in the clearest ocean waters. They form extensive beds or meadows, and these meadows can be found along every single continent except Antarctica. So here's a map showing you their global distribution. As you can see, they're found in polar, temperate, as well as tropical regions. The primary factor limiting their growth is light, but there are other factors such as nutrients, salinity, wind and wave patterns, temperature, as well as substrate type, which will either promote or inhibit where they grow and establish themselves. So seagrasses evolved somewhere between 400 and 100 million years ago during the late Cretaceous period. We know that they evolved from terrestrial monocots that reinvaded the ocean, and so they had to have a number of unique adaptations to allow them to live in marine environments. They needed to be adapted to salinity, they needed to be adapted to changes in carbon dioxide, as well as light, and they also needed to withstand the mechanical drag of water as well as the density. And here's just a quick figure showing you their evolution through time. Today there are 72 species. We classify them according to functional group rather than taxonomic group. And all species have a diversity of come in a diversity of sizes as well as morphologies. So sometimes you might find a species that's 2 centimeters in length, and other times you might find a species that have blades up to 7 meters in length. Morphologies include ovate leaves, as you see right here, fern-like leaves, then you have cylindrical leaf or strap-like leaves. But regardless of what the plants look like, they all have blades that are the primary site for photosynthesis, and they also have roots and rhizomes, which you can't really see right here, which are used to anchor them into the ground. So why do we care about seagrass meadows? They serve a number of ecological as well as economic roles in our coastal systems. First thing that they can do is stabilize sediments, and so this prevents erosion. They can also trap and cycle nutrients, and so this improves both water quality as well as clarity. They also provide food and habitat to a number of different organisms. Those organisms may be commercially important, such as tautog or striped bass, or endangered. So that would include sea turtles, seahorses, as well as manatees and dugongs. We say that they're an indicator of estuarine health, and the reason for that is that they are vulnerable to changes in environmental conditions, so they rapidly decline when environmental conditions decline. They're also considered a significant carbon sink, and this is important to mitigating global climate change. So even though they make up 0.2% of the ocean waters, they can store twice as much carbon per unit area as temperate and tropical forests. So this is very important. Here are some images just showing you different organisms utilizing seagrass habitats. The left top, we have periwinkles that are grazing on the seagrass leaves. And we have sea turtles that use beds. They actually eat seagrass. This is a native rock crab that forages inside seagrass habitat. We have squids, we have shrimp, sharks can be found in seagrass beds. You can also find coral fish, and this is a manatee on the bottom right-hand corner. So even though seagrasses provide a number of services in our coastal systems, they are facing a global crisis. We are losing 50 football fields each day to multiple stressors, and 14% of seagrasses are at risk for extinction. We have 3 that are currently classified as endangered. And this is showing you where they are declining in each of the world's different regions. We have 1 to 2 species declining in North America on both coasts, in parts of Europe as well. We're losing 3 to 4 species in the Mediterranean, in the Indo-Pacific, on the coast of Australia, and we're losing 5 to 9 species in Japan, as well as the coast of China. So what are causing these losses? The most documented threat is changes in water quality as well as clarity. There are other factors. In tropical systems, we have issues with changes in salinity. That's from irrigation systems. We also have issues with coastal erosion, and so you get pulse turbidity events. We have issues in tropical regions with urchin grazing. That's due to excess nutrients. We have issues with eutrophication, and for those of you that aren't familiar with eutrophication, that's when you have excess nutrients coming into the water. This causes phytoplankton blooms and algae blooms, and this reduces the amount available to seagrasses who are living below. They also have issues with dredging and boating activity in tropical regions, and what happens here is when you have boats going through seagrass beds, they can create prop scars. They can also uproot plants with their anchors. And mooring fields can shade plants, and so the seagrasses don't get the light that they need, and so they die off. In temperate regions, we have a lot of similar threats. We have issues with eutrophication. We also have issues with high water temperatures combined with low light. We have issues with disease. Back in the 1940s, we lost 90% of the North Atlantic population of eelgrass, which is a dominant species up here, due to wasting disease. We have substantial issues with herbivory from waterfowl, urchins, as well as turtles. And then we also have a lot of issues with non-native species displacing seagrasses. The important thing to note here is that anytime you lose a seagrass bed, you also lose any of the key ecosystem services it provides. So what are we doing to help us assess the status trends as well as health of seagrass beds? We have a number of monitoring programs going on. There are two major programs. There's Seagrass Net, which is based out of UNH, and there's also Seagrass Watch, which is based out of Australia. Both of these programs have global monitoring protocol, and they have web-based reporting. The participants include academics, citizen scientists, as well as managers, so pretty much anyone can participate in them. Seagrass Net has 126 sites across 33 countries, and Seagrass Watch has 259 sites across 17 countries. I'm most familiar with Seagrass Net, although I have participated in some Seagrass Watch programs. What we do in Seagrass Net is we go out and we find a bed that we want to monitor. We delineate the edges of the bed, so we find the shallow edge, the middle of the bed, and then the deep edge. And then we put in transects, permanent transects, 3 of them. And we come out anywhere between 2 and 4 times a year, and we visit each of those transects, and we lay quadrats. And those are just square frames. And in each one of those quadrats, we collect information on seagrass parameters, such as percent cover. We look at shoot density. We also look at species composition. We look for signs of disease, and we also look for signs of grazing. In addition to collecting seagrass parameters, we also set out sensors to look at environmental conditions. So Seagrass Net has been very effective in documenting changes as well as identifying causes. So back in 2005, we noticed the disappearance of a bed in Sabah. This bed happened to be in a marine protected area about 60 miles from shore, and so we considered it a pristine environment. When we started looking at the data, we found that there was 60% cover, so you can see right here, back in 2012, and then it started to decline somewhere in 2004, and then it was down to 0% in 2005. So we started looking at our sensors, and what we found is when we looked at our light sensors, that the beds were receiving in 2001 50% light intensity, but however in 2002 they were down to 30%, and then from 2002 to 2005 light intensity went down to 20%. And seagrasses need more than 20% of the surface irradiance to survive. So what we did was we started looking at aerial photographs and remote sensing imagery, and what we found was that they were developing the coastline, they were removing trees, and as they were doing so, there was a lot of erosion, and they were getting these massive plumes that were going out over 60 miles to our seagrass bed. causing it to decline and disappear during that time frame. So what do we do when we start seeing declines or we lose seagrass habitat? We restore. Restorations have been conducted since the 1930s, so this is not a new concept for seagrasses. You can either restore using seeds or you can use vegetative shoots. The first guidelines were published by a wildlife biologist here in Massachusetts by the name of Addy. He actually worked at the Parker River National Wildlife Refuge in the Great Marsh. And his guidelines were published over 75 years ago, but we still use a lot of his recommendations today. The first thing you need to do before you restore is you have to identify and remove your stressor. If you don't identify and remove your stressor, there's no way that you can expect your restoration to be successful. After you identify and remove your stressors, you need to identify donor populations to put into your restoration site, and I'll go into that a little bit more, but the general idea is that not all donor populations respond equally to transplanting. You also need to develop models to properly select sites within your system to restore, and I'll talk about that again in a second. And then the last thing you should be doing is spreading your risks. So rather than transplant all at once, you should probably look over a bunch of different seasons, as well as years when you're transplanting to maximize the amount that's established. We do restorations because we know that they increase the ecological and economic services within systems, but what a lot of people don't realize is that by restoring it also facilitates natural recovery. So for instance, eelgrass, again, which is a dominant species here, it takes about 100 years for a patch that's this big to grow to 1 acre just through vegetative expansion. The other benefit of doing restorations is that it raises public awareness, and so this is important. Anytime you have the public involved in your restorations, that promotes stewardship, and so people are more likely to preserve this natural habitat. Even though there are a lot of benefits to restorations, we do know that they're very expensive. It costs the state of Massachusetts approximately $100,000 per acre for the state to put in eelgrass. And then also, we have a lot of variable success rates. So right now, I'm just going to quickly go through a restoration I'm working on here in the Great Marsh. This is part of a larger project to increase the overall resiliency of the Great Marsh system. And if anyone's interested in that, I can tell you more about that later. So for those of you that aren't familiar with the Great Marsh, it's on the upper north shore of Massachusetts, and it consists of 2 major water bodies, which are Plum Island Sound in the north and Essex Bay in the south. Great Marsh is the largest wetland-dominated estuary in New England. It has over 20,000 acres of salt marsh, and eelgrass used to be abundant here in the early 1900s. We're not exactly sure why it disappeared. It disappeared somewhere around 1950. We do know that there was an outbreak of the wasting disease in the 1930s, so that probably impacted the population there at that time. We had over 40 acres back in the early 1900s. We also know that in the 1950s, eelgrass was starting to encroach on clam flats, and the clamming industry was taking off in that area, and so clammers were starting to remove eelgrass that was encroaching in the area. We also know that there was some watershed development, so there's probably excess nutrients going into the system. And lastly, we also know that there was an issue with green crabs. Green crabs are an invasive species. They arrived here sometime in the 1800s, but their populations have been going up and down since the mid-1900s. Green crabs were so abundant back in the 1950s that clammers had to put fencing up around the clam flats to protect the clams from predation. And we know that green crabs can come in and uproot eelgrass, so we think that they had a major impact on eelgrass beds. The reason why we're restoring Great Marsh today is that it's an ideal location for a restoration because environmental conditions are comparable, if not better, than other systems currently supporting eelgrass beds, and also because the area receives a lot of local, state, and federal interest. So we've had a project going on for about 4 years now. We started in 2012, and the first step of our restoration was to develop a site selection model to help us prioritize sites for restoration. So the first thing we needed to do was identify all the biological, chemical, and physical factors that promote the establishment and growth of eelgrass. So one factor we collected data on was bathymetry. We wanted to identify all the areas within our system with a bathymetry between 1 and 4 meters. Less than 1 meter, you run the risk of plants becoming desiccated if they're exposed during low tide, and more than 4 meters, you run the risk that plants will become light limited. We also collect a lot of sediment information. So eelgrass likes to grow in areas with less than 70% silt clay, so we needed to identify those areas. We had 15 years of water quality data for this system that we needed to process. We looked at total dissolved nitrogen, we looked at chlorophyll concentrations, we looked at TSS, and we looked at salinity levels at different stations throughout the system. We also collected water clarity information. We looked at the location of intertidal flats. Just in case we didn't pick that up in the bathymetry information, we wanted to make sure that we were not planting on intertidal flats where clambers would be. We looked at wave exposure. We needed to avoid areas with high wave exposure because that could uproot our transplants. We looked at information on historical eelgrass, and then we also looked at the location of moorings. We did not want to plant in areas where there were boats because they could shade the plants. So once we got all of these parameters, we threw them into our model, and our model rated all the areas within the system. So anything that had a rating of 0 or 2, which is denoted in red, we said wasn't suitable. Any area that was in orange received a rating of 4, we said was suitable. Anything with a rating of 8 or 16 is considered very suitable or most suitable, and those are in yellow and green. And so in the end, we had about 1,300 acres that our model was telling us was good for a restoration. But rather than just going out and doing a full-scale restoration the next 2 years, we decided to test transplant just to validate our model. So before putting in 3 acres or 5 acres, you would just want to take a few plants in a number of different locations and see whether or not they survive. So we started test transplanting at 24 sites within those 1,300 acres, and we used a variety of different methods. We used something called the horizontal rhizome method, where you take, it's pretty much like similar to gardening. You dig a hole, you take 2 plants, you stick it in. We also use the washer method, which is you tie plants through a hole, washer, and you disperse them. We use biodegradable frames. One thing that we did that was different was that we used multiple donor sources. This is a new concept because we've found in mesocosm studies and field research that not all populations respond equally to transplanting. And we also wanted to build a genetically diverse population because we know that genetic diversity increases resiliency. So today, this is my second to last slide, we are doing our full-scale restoration. We have identified one area in Essex Bay that transplants extremely well. We are putting in 3 acres. We're using the community to promote stewardship. I work with New England Aquarium, high schools, Student Conservation Association, and a number of different organizations. We plant throughout the year from May to November to spread our risks. And we also have a green crab management program because there are issues with invasives. We're monitoring at this time. We've got good establishment of the bed that we've transplanted so far, and we're also assessing ecological function. And I think what some people will find that's very interesting is that after 3 months of transplanting, we have 90% of the organisms in our restored bed that you would find in a natural bed. So that's excellent. So in summary, I just hope everybody took away that seagrasses are a great ecosystem. They are facing a global crisis. They're declining everywhere. There are multiple stressors responsible for declines. And monitoring is one great way to collect detailed information on their health status and trends. And restorations can help to mitigate for loss. So thank you. I did have a video, but it's not working. So I'm sorry.